Thank you very much, Matt, and thank you very much for the invitation.
Delighted to have you.
Thank you very much, and I apologize I could not make it yesterday. I missed a very wonderful dinner, so apologies for that. So I'm from the National Center for Computational Toxicology in Research Triangle Park. I work for the EPA, and I'm going to be changing gears a little bit and talking a little bit about high-throughput methods. And as Matt pointed out, the challenge that we're trying to face is the issue of having many chemicals and not enough bioactivity data to be able to characterize their health effects and figure out which chemicals are really the ones that are bad actors, and if the ones that are bad, how do we actually figure out a point of departure for them. So with that, let me just give a quick background. Since this is the liver symposium, we have to talk about liver toxicity, and this is an analysis of the integrated risk assessment system at the US EPA, in which basically this is what the EPA uses for human health risk assessments internally, and this is where a bunch of human health risk assessments are kept, if you're all familiar with it. About 20% of the chemicals in here that have been evaluated so far, and there's not a lot, only 500 or so have been actually systematically characterized for human health risk. And 20% of them have been, the critical effects are really based on the liver. So the liver is actually pretty important insofar as environmental chemical risk is concerned. If we look at all the guideline testing data that are available out there currently, and they've been stored in this database that's been developed by one of my colleagues whose name is Matt Martin, and the database is called ToxRefDB. It's actually publicly available. You can go to our website, download it. It has more than 5,000 guideline testing studies, acute, subchronic, chronic, and there is a massive amount of information on hepatic histopathology in rats, mice, rabbits, dogs, all kinds of animals. And what we've done is try to summarize basically the different grades of liver injury that we see, more than hundreds of histopathological lesions, and try to categorize them into some sort of summary scheme. What we find is that you see a whole bunch of types of lesions that have to do with aging, that have to do with chemical-induced hypertrophy, steatosis, various types of injury, cholestasis, inflammation, and then the progression from pre- and neoplastic lesions all the way to neoplastic lesions. So since this is a liver crowd, I thought I'd put up some hepatic histopathology information. We're using this, by the way, to mine associations with high-throughput screening data. And that is not really what I'm going to be talking about today. Today what I'm going to be talking about is this whole idea of how do we actually use some of these high-throughput data streams to begin to think about the deviation from normal function and how you actually move toward toxicity. And how can we use unique data streams that we're generating out of the ToxCast project at the EPA to be able to understand that. And what I thought I'd do is put up a figure from a paper by Mel Anderson, Krusky, and others who have discussed this idea that when you have environmental exposures or chemical exposures, you observe this. Of course, your chemical has to go and reach its target organ cells, and inside the cells it brings about some sort of biological interactions. It brings about some perturbations, and those perturbations alter cells. Those alterations in cells, as we heard about, there are various adaptive responses within cells, NRF2, as well as others, that try to counterbalance those changes. And if those changes cannot be counterbalanced, you begin to see cell injury, and you can see all kinds of other lesions forming in vivo. So what we're trying to do is, in computational toxicology and high-throughput sort of screening and all this, think about toxicity across this continuum of changes that happen. When you are exposed to environmental chemicals, you get the distribution of the chemical down to the tissue, down to the organ, into the liver, for instance. You see changes in molecular targets, you see cellular changes, you have diverse types of cells that interact in complex networks. those networks get perturbed, and ultimately you see toxicity. So what is it that we do in the CompTox Center in computational toxicology? Well, one of the things that we do is we have a battery of hundreds of high-throughput screening assays that measure cell-free activities, cell-based activities, transcriptional activation activities, other signaling pathways. And we try to assemble that information from different cell culture systems, and then we use computational predictive models and try to figure out whether or not we can identify markers or whether we can identify critical concentrations at which certain chemicals cause certain effects. So today I'm going to be talking very specifically about one of those platforms that has to do with high content imaging. This technique has been used for a long time of course, in biomedical research. And we've used this platform in ToxCast to look at all of the chemicals, about 1,000 chemicals. This particular assay is not in a very exciting cell model. It's in HepG2 cells, but these cells have actually been the workhorse of toxicology in chemical testing for a while. We've used HepG2 cells. There are 10 multiplexed assays, and I'll talk a little bit about them. We took about 967 chemicals. Each chemical was, the cells were treated with about 10 concentrations of the chemicals, ranging from about 0.4 micromolar to 200 micromolar. The treatment durations varied, of course, all the way from 0 all the way to 72 hours. So we have not just dose, but we also have time. We have replicates. And just to get a sense of the data, we have about 10 to the power of 5 wells, hundreds of plates. In each plate, we actually, there are these fluorochromes in there that label various proteins and membranes. So there's stress pathway activation, there's mitochondrial function, there's cytoskeletal architecture, there's oxidative stress markers, there's p53 activation, there's, of course, nuclear labeling that lets you figure out what the state of the actual cell is and what the nuclear changes are. So with this, we actually get a massive amount of data, as you can imagine. We have 6 images coming out per well, 4 channels per image, about 500 cells or so per image. And that becomes a very substantial amount of information. So to be honest with you, for a year or so, we were just sitting on this data and didn't know what on earth to do with it because it seemed to be just so massive and so difficult to grapple. But then we started looking at the information, and just to give you some background, very, very basics on what this high content imaging data, what these data actually look like. So one of those images out of one of those wells for one of the control chemicals, which is DMSO, this is basically showing Herc stain on the raw image on the left-hand side. After some intensity analysis algorithms and object identification algorithms, which all they do is basically they look at the intensity and they try to find the nuclei, and then those nuclei are segmented out, and the algorithm then knows that's where a specific cell is. With that, you can actually build an DNA content like histogram and do all kinds of other analyses. So one channel has this nucleus stain. The other channels in this particular plate had phosphohistone H3. And this is the actual intensity image from that particular channel. This image over here is showing you the actual identification of the overall cells and the distribution of the stain through this channel. This image over here shows you a combination of the nuclear channel where the nuclei are and a combination with this cytoplasmic channel, so you can actually figure out what was the intensity of this particular marker in the nucleus versus the cytoplasm. Now, these algorithms are not 100% accurate, of course, but they do a reasonably good job of figuring out what the distribution of, how much of a particular fluorochrome is present in a particular cell. And you can actually begin to make assessments of what the actual state of the cell is in terms of the activity of a particular marker. This is a slightly different marker. This is mitotracker red, as you all know. This shows you, this measures mitochondrial depolarization. So if you have mitochondrial membrane potential changes, you're going to actually see an increase in mitotracker red. And that you can also go ahead and measure in different ways. In our assay, we actually had two ways of looking at mitotracker red, looking at the overall intensity distribution per cell, as well as looking at the texture of the mitochondrial membrane potential to be able to see what the actual structure of mitochondrial mass was. So do you have more mitochondria being formed? Are you getting some sort of mitochondrial biogenesis or mitophagy? Or how do you distinguish from mitochondrial membrane potential changes? We also looked at phosphotubulin to look at the cytoskeletal architecture. So for each well, each image analysis like this was done. And fortunately, we didn't do all the analysis in-house. We just looked at a subset of the data. This experiment was run by a company which was originally called Solumen, then became a Predica, and now is owned and called Ciprotex. And we looked at a total of 10 endpoints. We looked at the nuclear stain, from which we were able to look at nuclear size distribution changes, cell cycle arrest changes, cell number changes. We have mitotracker red that allowed us to look at the overall mitochondrial mass, as well as the mitochondrial membrane potential. We had phosphohistone H3, which is a marker of mitotic arrest, p53 activity. Tubulin, which tells you about microtubule organization. C-Jun phosphorylation, which is again a marker of stress kinase activation. And H2AX, which is one of those tricky markers, which is a DNA damage marker. But the question is whether it's actually looking at DNA damage in this particular model, or whether it's just a surrogate for some level of oxidative stress. So what do we actually do with data like this? Well, if you look at the results that you would obtain after you analyze the data and do this sort of analysis of treated versus control, you can get your traditional sort of heat maps or concentration response curves. So you can figure out what the AC50 value is, or some lowest effect concentration is for a given chemical at a given time point. So here's the result that you obtain for microtubule, the microtubule signal. So you see some sort of dose-dependent increase at 72 hours. You see some dose-dependent change in mitochondrial mass. But the biggest thing, since this is going to kill off the cells, you see a massive change in cell number. So you see a reduction in cell number, which is signified by blue. So just to clarify, red means something is increasing, and blue means something is decreasing. Yellow means it's not changing. So, okay, so that's very interesting. But then you've got 3 time points and multiple chemicals. And if you want to look at the effects of an individual chemical, you get actually quite a bit of interesting information. So here's what we get for the different time points and the different concentration, for the concentration response across these 10 endpoints for butachlor. So you see changes in p53 activation, you see changes in cis kinase activation, you see changes in mitochondrial membrane potential at higher concentrations, you see changes in the cytoskeletal architecture. But the question that is hard is, when you've got about 1,000 chemicals, what are you going to do with that information? You can write an interesting paper about all the possible AC50 values for each chemical, and that's interesting. But we've got some very interesting time course data in addition to dose response data. So we can summarize it and say, well, overall, across all of these chemicals, you see that. So this is a summary in terms of pie charts showing you the number of chemicals that at any given time point produce significant changes in a particular endpoint, increases or decreases. So you can see here that mitochondrial membrane potential across was decreased for 28 chemicals out of all the chemicals that we studied. And at 72 hours, the largest change that you saw was a decrease in cell number, which is what you would expect overall. But in between, most of the chemicals changed multiple endpoints. So there is some complexity in terms of understanding the biology, and we're trying to build models to do that. But one thing we thought we might look at first, and from this notion is this whole idea of looking at the response of the system in some dynamic way, was to look at some of these. So if we look at all the chemicals, about 1,000 chemicals, 10 concentrations, 3 time points, and 10 endpoints, if we look at all of these data and we just collapse it down, so one approach for doing that is a clustering approach called k-means clustering. And there are many ways in which you can, of course, summarize this. This is one approach for doing this. What this shows us is that you can collapse all of these data, which is about 30,000 responses, profiles of responses for this HepG2 system. And you see this, that all the responses can be summarized in this case by these 15 characteristic profiles. And let's assume for a second that they correspond to, and you can use different approaches, by the way, and most of them produce a similar profile of activity. So let me explain what this really means. What this means is that most of the chemicals, this bar graph on the right says, most of the chemicals produce pretty much no effect. So these circles correspond to how many chemicals brought about the changes, how consistent were those changes. When these circles are blue, it means that most of the chemicals in that particular group decreased that endpoint, which is given in the column. If you see a red circle in a place, that means that most of the chemicals increased that particular endpoint. As you go up over here at this particular state, you see this big blue circle corresponds to those set of chemicals that consistently and potently and efficaciously decreased mitochondrial membrane potential, is this particular group over here. And so on and so forth. This group up here is the group of chemicals exposures that produced massive changes in p53 activation, stress kinase activation, oxidative stress, microtubule changes, and ultimately decreases in cell number. So I'm going to ask you to sort of suspend disbelief now and follow this sort of analogy a little bit. So let's consider that this is a summary of the phenotypic state of the HepG2 system. And that all chemicals at different concentrations at different time points, when you hit the HepG2 system, they land somewhere in terms of this phenotypic structure of the system. So here's an example. I'm going to walk you through one chemical at one concentration. And let's say starting at 0 hours at this concentration, the state of the HepG2 system falls into this particular profile of activities across these endpoints. This is the 0 hour response. At 1 hour after treatment at this concentration, what we observe is that the activity of the system can be summarized in terms of this particular phenotypic response, in which you have a little bit of increase in p53 activation, you see some stress kinase activation, you see a little bit of mitochondrial membrane depolarization, and not a lot of cell loss going on. If you continue and look at the system at 24 hours, what you see is that the system actually seems to recover, and the activity of the system has actually changed, and it's come back to this lower level of overall activity. And if you look at 72 hours, you actually see that the system actually returns to its normal state. So if you now ask the question, what happens if I look at every concentration of this particular chemical and see how it perturbs the system? What you observe is this pretty interesting view, which what we call is what we define as trajectory. So a trajectory is basically the sequence of states that the HepG2 system follows after it's perturbed by a certain concentration of the chemical. So in this particular case, at the lowest concentration of 0.39 micromolar, this particular chemical produces this sequence of states. It starts from this particular normal configuration. There's perturbations, it goes up to this particular state, and then it returns and recovers at a later time point. So this is time 1, 2, 3, 4, and this is going from 0 hours to 72 hours. As you continue to increase the concentration, you see that the perturbations tend to either increase, and the level of recovery that you see in this trajectory is not complete. So as you increase the concentration further and further, you actually see that the system gets more and more perturbed and doesn't actually recover. So with this notion, when we actually looked at hundreds of chemicals like this and found that what you could begin to explain these results in terms of is that at certain concentrations, what you're observing is the trajectory of the system is showing recovery. But then at some concentrations, you're not seeing full recovery. And as you keep on increasing the concentration of the chemical, you don't see recovery at all. So with this idea comes the notion that perhaps there is some dose-dependent transition that happens. And at that point, beyond that point, the system doesn't actually recover to baseline. So we tried this idea and looked at not just this chemical, but hundreds of other chemicals. That we have 967 chemicals. So, it could be that you could observe this by chance for 1 or 2 or 3. But if you start seeing this pattern again and again, then perhaps it means something. So here's another chemical called butachlor. And butachlor produces a similar sort of profile. But in this particular case, as you see, the characteristic change in state or the trajectory of the system is different at different concentrations. At lower concentrations, it does not produce as many perturbations as the previous chemical fluazinam did, and it doesn't actually produce the full range of perturbation that you see either. So the trajectory is a little different. And so is the tipping point. So the tipping point is some point right in between these two concentrations, after which you don't see recovery of the system. So you can look at other chemicals like troglitazone and other pharmaceuticals. We actually have a whole bunch of failed pharmaceuticals in this set as well that we've looked at to see, does this really begin to explain these results? Now, troglitazone, for instance, is, as you all know, is a well-known anti-diabetic agent that's been prescribed, and troglitazone was removed due to liver injury. And if you can see, troglitazone actually produces interesting perturbations. It does produce recovery at lower concentrations, but it does produce a lot of oxidative stress in this particular model in HepG2 cells, and that's been described. It's not something new that we found. Troglitazone produces effects in HepG2 cells. And here's the other chemical that actually was found to be safer for humans and actually is currently used. It's pioglitazone, and this does not produce much perturbations, and up to a certain amount of concentration, and you can see that it produces complete recovery. It's not until very high concentrations that you don't see recovery of the system. So with this notion, what we thought is, well, maybe we can use a little bit of math and do some more quantitative analysis of the data and try to figure out in a very quantitative tipping point in terms of real concentration-dependent transitions. And what we found is that you can actually do a little bit of math and calculus and modeling and figure out that there is actually a critical concentration at which the system will actually not show recovery. And that you only observe that critical concentration or tipping point for some chemicals and not for all chemicals. And if you actually analyze all of the chemicals, this is a summary of all of the chemicals that produce tipping points. And let me quickly describe what this is telling us. Each of these points over here, each row is a chemical. Over here, what you're looking at is that critical concentration. These are the chemicals that produce the lowest critical concentrations, and these are the chemicals that produce the highest concentrations. There is noise, there is variability, so we have some uncertainty over here, of course. But the interesting point to note is that this critical concentration is generally much lower than the concentration that produces significant cell loss. It's hardly ever above it. So the cases where cell loss is greater than this critical concentration is these situations over here where you see these red dots or green dots to the left-hand side of this particular distribution. On the right-hand side here, you're looking at actual LEC values for that chemical at the 72-hour time point, and here you're seeing some other statistics associated with that. So this was a pretty interesting result, and we've actually submitted this. It's currently in review, and we're trying to see how to move forward with that. But the real question that it opened up for us in looking at this was, well, you know, we've always thought about toxicity in terms of adverse histopathologic outcomes or real adverse bad things happening, and that's true. I mean, there has to be something real hard phenotypic clinical that you can anchor towards. But if you're trying to look at regions of safety for chemicals, perhaps we can think about these tipping points as valuable as a valuable framework to begin to analyze the data and look at the data. And one way to think about is in terms of trajectories is that the normal system has a range of normal functions and has a range of normal trajectories. And when you hit the system, expose it to a chemical at a certain concentration, you're going to see all kinds of perturbations and some cellular changes. In many cases, the system will have enough homeostatic capacity and adaptive capacity to be able to recover to its ground state. But in some cases, the system will not recover, and you're going to see basically some sort of irreversible injury. Now, of course, the system that we used is limited. It's HepG2 cells. We only looked at 10 endpoints, and we only looked at a 72-hour time course. But given the number of chemicals and given the phenotypic relevance of these endpoints, because these endpoints are not just changes in gene expression or changes in small numbers of genes, these are actual measures of cell health. They look at very, very key cell state changes that actually inform us provides valuable information about whether the cell is injured or not injured. So the question is, we need to extend this analysis. First of all, we can't just use HepG2 cells. So one of the things that we're beginning to do is work with folks that have very complex co-culture or multiculture systems that have been derived from stem cells, in which we can actually begin to model. Sorry, that's my own timer telling me 25 minutes are up, so I should wrap up quickly. And I just have a few more slides left. So we need more physiologically relevant models, and of course we need more sophisticated computational models, because even if we do generate such data and such trajectories in vitro, extrapolating that to actual scenarios where we care about human health requires larger timescales and need to consider ADME and dosimetry and all kinds of interesting questions. So one of the things that we're doing, and EPA has invested quite a bit in this very recently, and there'll be more information becoming available about this in the coming months, we're going after organotypic culture models. Organotypic culture models are these bioengineered complex cultures that can be derived from stem cells, but one of our collaborators, in fact, has used HepG2 cells and grown them in a 3D culture and found that, first of all, their liver phenotype tends to return. They start looking like liver cells again, surprisingly. They start producing enzymes that you don't see in a monolayer culture. So that's very interesting. And also, we're beginning to look at these sort of vascularized, you may have seen this paper in Nature, looking at these liver buds that were produced from stem cells. You can actually, groups are actually vascularizing these and coupling them to microfluidic devices to begin to look at actual flow into them, and to measure flow into them, and to use multi-resolution imaging and live cell imaging to generate not just these real cross-sectional data sets, but real longitudinal data sets that can tell us about dose, time, and multi-dimensional responses. Finally, one of the reasons why Matt invited me over here was to talk about virtual tissues and microphysiological systems that we can actually simulate computationally. That's one of the things that we've been doing for a while. And our idea basically is that we take traditional physiologically based pharmacokinetic models in which you translate exposure into internal concentration. We couple that, we've actually published approaches with which we can actually estimate with a considerable level of accuracy what the cell level concentration is of the chemical given an oral exposure. And we can go into individual cells and use systems biology approaches to model different types of mechanisms that we care about. One of the mechanisms that one of our collaborators at the Hamner is using is the NRF2 model, in which they're looking at adaptation and the activation of NRF2 and how that leads to adaptation, and we're hoping to be able to couple that model into this. And with that, what we can do is we can actually begin to simulate a small prototypical lobule in computationally. And we've published some preliminary results on this, but of course there's a long way to go. We need much more data to be able to calibrate and evaluate such a system. And what we intend to do is look at how resilient the system is, whether it recovers. Originally we were hoping 3-4 years ago that we'd be able to begin to simulate hepatic lesions, and that's maybe a possibility down the road. Right now we're thinking about just looking at resilience, recovery, and looking at the trajectories of this system. So with that, I'll just summarize and say that EPA ToxCast project is, as you may have heard about, it's this framework for looking at high throughput testing chemicals. We have hundreds of 600 or 700 assays, cell-free, cell-based, different types of aspects, a lot of, many liver assays, by the way. We have about 1,000 chemicals. This analysis was just based on the high content imaging aspect of this data set, which is actually quite informative and useful. The most interesting part of this set is that it's got time course in there, and time course is actually very valuable to begin to look at some of these points of departure. If you just look at a single time point, you can't see these things. You will miss these things. And the notion that we're going after right now is that you can use these time course dynamic data sets to begin to map out system trajectories. From these trajectories, you can begin to see what the tipping points are, and from those tipping points, maybe you can come up with points of departure. That's something that, again, we're working toward seeing whether it's feasible or not, and we'll see where that goes. What we do need is more physiologically relevant models, and organotypic culture models that are out there right now are actually becoming quite amazing at what you can actually measure in them, And you need computational modeling and simulation to be able to put this information together and make predictions or extrapolations from in vitro to in vivo. So with that, I'd like to acknowledge the team. There's a lot of people that did a lot of work. The high content experiment was originally designed, conducted by Keith Hauk, and David Dix, who used to be with the center, is now in OCSPP and involved in the endocrine disruptor screening program. Predica Ciprotex is the company that ran the assays. My colleagues Matt and David were involved in data processing. Postdoc John Jack was involved in the initial analysis. My grad student, Ji Lee, has been involved and currently involved in data processing. Woody Setzer has done a lot of the statistics and concentration response analysis. And this notion of trajectories and tipping points is something that we've batted around internally a lot. Rusty Thomas, who recently joined us as our new director, has been very, very closely involved with this. Richard Judson, of course, Tom Knudsen, and Bob Kavlock, who used to be our director and now has moved off and is the deputy director for assistant director for science at the Office of Research and Development. So with that, thank you very much. I'll take any questions. Are you, or shall I? Okay. Go ahead.
I'm going to talk a couple questions regarding your high content imaging experiments. Number one, I assume that you also looked at chemicals that you already had in vivo toxicity data. How well was false positive, more importantly, false negative predictive value?
So we are currently we did not phenotypically anchor the HepG2 data to the in vivo data. So right now, in fact, we have a second analysis that's looking at a subset of about 320 chemicals that were run in primary rat hepatocytes. So what we're doing is taking the primary rat hepatocyte high content data, which is slightly different endpoints but similar, and we have the guideline testing results from subchronic and chronic studies, and we have in vitro pharmacokinetic data that looks at plasma protein binding and intrinsic clearance. So we're trying to put those pieces together and look at them. Right now, I can't give you a very good rate of false positives and false negatives. What I can say is that the point of departure that we find out of this is generally always lower than whatever LEL value or LOL value that we find. So we do a pretty good job with that. Sometimes we're over predicting it, but I don't have an exact number for you right now, so I apologize.
Now, the other question is, one of the beauties of high content imaging is you also get data at the level of the individual cells. So when you see these changes in the well level, is that, all the cells are decreased 50%, or is it 50% of the cells are decreased 100%? What's the pattern?
So that is a very interesting question, and unfortunately, because there is so much data, we couldn't delve into that deeply. It's very clear that most of these endpoints, if you actually look at the concentration response profile, it's very non-monotonic in some cases. And one of the reasons it's non-monotonic is because you have different subpopulations of cells. And so a few of the endpoints like mitochondrial mass and cell cycle arrest are very dependent upon the subpopulations and how they're responding. So it's a completely different sort of set of questions that we have to ask. What we're hoping to do is use some of our computational simulation approaches to begin to see how the cell-level behaviors and cell-level phenotypes impact the predictions that we make. But there are a lot of subpopulations that do very interesting things.
I had a similar but slightly different question. Have you ever correlated your point of departure data with cell death? I'm curious if
Yes.
you get a correlation.
In this?
Yeah.
So yes, and there generally we are much lower than the concentration that produces cell death.
Whether you're lower or higher, but whether there's a correlation.
No, it's not a very high correlation, and the reason for that is that in this assay you don't actually get a readout if there's a lot of cell death. So what we're correlating, so you don't get cytotoxicity related cytotoxicity, because if you don't see a valid cell, it doesn't count it. So you don't get that confounding until you go to more than 200 micromolar.
And so my other question was, the HG2 cells have an obvious advantage of being a very simple system. But as you talk about going to more complex systems like virtual tissues, it seems to me the cost and the labor is going to become very high. And at what point do you say, let's go buy a mouse?
Is there anybody else who'd like to answer that question? So that's it. Each of these studies cost, so I can't give you the cost of the study, but we're talking about a few $1,000s to generate the profile for a single chemical versus a guideline testing study. So it still is going to be much cheaper, no doubt. Even the complex co-cultures that you heard about, there are approaches for actually, the whole idea of EPA funding these centers is to make it high throughput. If it can't be done in high throughput, then it's not really worthwhile following it. So it is going to cost much less. And I know the EPA policy is not about, we're about high throughput.
I think my question is somewhat related. I'm trying to understand when you do this system trajectory that you look at the recovery, right? Whether you have full recovery, partial recovery, or no recovery. So the way that you calculate and you determine the recovery, is that the sum of all the endpoints that you measure, or is it just whether the cell survives or dies?
In this particular case, it is the total, all of the endpoints that we register. It's not just the cell dying or dividing. So it's a aggregated measure of all of the endpoints that are showing perturbation. So think about it in terms of deviation from normal. So the more there is the deviation from the normal background, the greater the perturbation is. And the lesser that there is the deviation from the background, less the perturbation is. So the recovery is that it returns to its normal configuration based on these endpoints.
Technically, a cell that you could define a cell that has recovered as a cell that is alive but still has a number of liabilities, P53 positivity, some mitochondrial liability and so on, but still the cell is alive. Is that what?
So to be very honest, this is based on a cell population. The trajectory is based on the population of cells. And it's not based on an individual cell because we have snapshots in time. So we can't technically track each cell to see whether the actual things change.
But the bottom line is that at the end, where the cell is based on the sum of all the endpoints that you measure.
Yes.
Not just
whether it's
cell that is dead
no,
or alive.
no.
So what does it mean when your tipping point occurs at a smaller, larger concentration than your cell loss?
I thought someone might ask me that, and I was trying to think about a good answer for that, and I don't have a very good, satisfying answer for that right now. I'm not sure right now of the answer of that. I think the case where it's lower, I can explain that because it's more sensitive. Where it's higher, honestly, I don't know if it has I wish I had a good answer. So you got me. That was a tough one. I don't have a really good answer. Do you have an answer?
Well, you wouldn't like my answer.
Maybe you can tell me later. Let me save some face then.
Time for one more question. Dr. Barbe, did you have a question?
Taking a look at all these endpoints, moving forward, how complex or relatively easy it's going to be to develop coordinated causal relationships with different endpoints that are tuned together?
So I have a postdoc who just started about a month ago who's actually trying to take the dose time data to put it, at least for these 10 endpoints, into a causal model. And just to be clear, we actually have a number of other assays in HepG2 cells, like multiplex transcriptional reporter assays that are based on a number of transcription factors, about 70 of those. So we're trying to see if we can piece it together with those, where we've got NRF2 as well, by the way, and a number of other CAR, PXR, and all the nuclear receptors as well in there. So one of the ideas is to really bridge that and pull that. It's just a question of time. That is one of our objectives.
That's highly relevant because it identifies therapeutic targets to those relationships.
Absolutely. That's a very good point. Yes.
I guess I had one question, one comment, then we'll go to the next speaker.
Yes.
So have you gone back and see if the cell model, did it how did it compare against, say, ToxRef DB or SEB, so chemicals that you might predict have hepatotoxicity? If those chemicals were in these rodent databases, how did that fare? Have you done that yet?
Yes, so I think I answered that partially before, but the cell number endpoint is highly correlated with hypertrophy. Mitochondrial changes are really highly correlated with hypertrophy in the 2-year as well as subchronic rat liver outcomes.
Okay.
So they are really correlated with the in vivo endpoints, and they come up as really good markers, predictive markers in those models.
All right. Well, thank you. So it's always amazing when I interact with EP